Leaves, leaves! It all happens in the leaves. Photosynthesis and cellular respiration are the bomb. Now listen to Sheep!
A leaf changes light energy from sun to chemical energy usable in the cell by using the chloroplasts, which contain chlorophyll, to convert energy from the sun into chemical energy that is usable in the cell. The chlorophyll is a pigment which has two variants, chlorophyll A and chlorophyll B. They absorb light and use that energy in conjunction with carbon dioxide and water to produce glucose and oxygen. The light reaction occurs in the thylakoid membranes. Light is used to drive the production of ATP and NADPH. Water provides the electrons needed and is converted to oxygen gas, a waste product. Photosystems are consist of complexes of protein and pigment molecules that are embedded in the thylakoid membrane. They direct incoming photons into the reaction center where chlorophyll A molecules produce excited electrons which are transferred to an electron transport chain. There are two types of photosystems. Photosystem 2 is the central chlorophyll that works best at a light wave of 680 nanometers. It is found at the start of the electron transport chain. Photosystem 1 is the central chlorophyll that works best at light wavelength of 700 nanometers. It is found at the end of the electron transport chain. Since chlorophyll is not going to have any electrons to return to it, new electrons are needed. Water provides the other electrons. This creates 4 protons and 1 molecule of oxygen gas for every 2 water molecules consumed. The oxygen gas is released as a waste product, becoming a major input for aerobic cellular respiration. Electrons move from photosystem 2 to photosystem 1 via the electron transport chain. From photosystem 2, they are transferred to the enzyme NADP reductase, which uses them to reduce NADP plus into NADPH. It produces both ATP and NADPH, and it also requires water. Electrons move from photosystem 1 to the electron transport chain before returning to photosystem 1 in the cyclic electron flow. It only produces ATP, but it does not require water.
Using carbon from carbon dioxide, energy from ATP, and high electrons from NADPH, the Calvin cycle constructs an energy-rich sugar molecule called glyceraldehyde.
The equation for photosynthesis is 6CO2 plus 6H2O with the addition of light energy yields C6H12O6 plus 6O2. Photosynthesis takes place in the chloroplast, in the chlorophyll.
All respiration begins with glycolysis. In the process of glycolysis, glucose is cleaved into 2 molecules of pyruvate with the usage of 2 ATP molecules and 2 NAD+ molecules. Products of reaction yield 2 pyruvate molecules, 2 NADH molecules, and 4 ATP molecules.
Acetyl-CoA enters. Acetyl group attaches to the oxaloxetate, forming citric acid. The carbons from the acetyl group are oxidized into CO2. 3 molecules of NAD+ are reduced into NADH. 1 molecule of FAD is reduced into FADH2. 1 ATP is produced.
This is a structure of a mitochondria. Here's a ribosome. The squiggly things are the cristae. The matrix is right there. ATP synthase particles right there. There's DNA. The outer membrane, the inner membrane, the intermembrane, and then these are the granules.
The reduced electron shuttles NADH and FADH are oxidized at the electron transport chain, complexes of protein embedded in the folds of the inner mitochondrial membrane. Electrons flow through the proteins in the chain, driven by the increasing electron negativity of the members of the ETC. As the electrons move through the chain, the free energy they release is used to pump proteins through the ETC proteins from the matrix into the inner membrane space. Oxygen serves as the terminal electron acceptor. When oxygen acquires 4 electrons and 2 protons, it is converted into water, which is released as a waste product. The oxidized electron carriers are fed back into glycolysis and the citric acid cycle. How ATP is produced. The ETC establishes a proton, aka electrochemical gradient. Protons can only diffuse back into the matrix through a proton channel. ATP synthase, the only proton channel available in the inner membrane. As protons diffuse through ATP synthase, the free energy that is released is used to catalyze ATP formation from ADP and free phosphate groups.
Fermentation is the chemical breakdown of a substance by bacteria, yeast, or other microorganisms. There are 2 main types of fermentation. Aerobic fermentation uses oxygen. Anaerobic is the respiration without the presence of oxygen. Aerobic produces 36 to 38 ATP molecules, while anaerobic only produces around 2 ATP molecules. Both occurs in the cytoplasm, but aerobic also occurs in the mitochondria.
Ethanol fermentation is a process in which pyruvate is converted to ethanol and a molecule of CO2. An example organism would be yeast. Lactic acid fermentation is a process in which pyruvate is converted to lactate. An environment in which this occurs are muscle cells. Photosynthesis changed the landscape of primitive Earth because plants were more abundant and they took in CO2 for when the Earth's atmosphere was rich in CO2. Plant evolution caused the CO2 levels to fall and the oxygen levels to rise. This made it possible for oxygen-breathing animals to inhabit the land. They were mostly amphibic.
Chloroplasts and mitochondria have their own DNA, which is different than the DNA stored in the nucleus. This makes sense since they would have needed their own genetic material when they were independent organisms. Today, though, much of the genetic code controlling mitochondria and chloroplasts' production and activity has been incorporated into nuclear DNA.